Jimmy, thanks as always for inviting me and giving me the opportunity to present. So this year I decided to present on Candida auris, which I think is really timely because we've seen a major change in the epidemiology again in South Africa. Candida auris, unfortunately, post-COVID is now the second most common cause of candidemia in South Africa after Candida parapsilosis. We've also got some new diagnostic and screening assays available. There are new antifungals, mostly in the pipeline. And I'll also tell you a bit about our new strategies for surveillance for Candida auris. I'm going to start by telling you, just to bring it back home, tell you a bit about an outbreak. So a few years ago, I was contacted by a clinical microbiologist at Chris Hani Baragwanath Academic Hospital, which, as most of you will know, has a very large neonatal unit, 185-bed neonatal unit. And I heard then that 10 babies with bloodstream Candida auris infection had acquired this infection over 3 months since June 2019. And of these 10 babies, 6 had died. All were in the surgical section of the neonatal unit. They closed the unit to outside hospital admissions. They instituted infection prevention and control measures, environmental cleaning and disinfection. It screened contacts, collected environmental swabs, all of which were negative. And this, of course, was of great concern. So Candida auris is a relatively new pathogen. Based on the phylogenetic analysis, it seems to have emerged probably in the last few hundred years. The drug-resistant strains of Candida auris probably only in the last 40 years. But really, it was only first detected in around 2009. It's now globally distributed across 6 continents, and as we know, behaves like a multi-drug resistant bacterium in many ways. Again, as most of you know, the WHO issued its first fungal priority pathogens list in 2022, and appropriately, Candida auris is listed as a critical priority pathogen alongside the other major human pathogens, Cryptococcus, Aspergillus, and Candida albicans. So throughout this talk, I'm going to use the simple conceptual model for Candida auris transmission in a healthcare setting. But before I get started with the meat of the discussion, just to talk about our laboratory-based surveillance for Candida infections and colonization in South Africa. So we use the germ surveillance network, which is functional across or operational across many hospitals in South Africa. Essentially, we depend on routine data which comes through the laboratory systems, and essentially routine specimens are collected by clinicians, processed by the routine microbiology laboratories to identify culture-confirmed cases. And these data are then reported through to NICD along with Candida isolates and detailed clinical data as well as additional data where possible. So let's start in terms of this conceptual model. We know that both infected and colonized people are infectious. They can then transmit the infection onto susceptible hosts either directly through the contaminated hands of healthcare workers or via a contaminated environment. So let's look at who is susceptible. So when do you think fungus, or when do you think Candida auris? Candida auris infections usually occur in a healthcare setting, either an acute healthcare setting or long-term care facilities, and usually among people with some sort of underlying disease or medical condition. So people who are severely immunosuppressed, for instance, people with underlying medical conditions such as diabetes, premature newborn babies, and then, of course, interventions. People who have received broad spectrum antibiotics, are being mechanically ventilated, have had abdominal surgery, for instance. And then, of course, COVID-19 is an interesting risk factor in addition to all of these. The pandemic, of course, created certain conditions which increased the risk of severe bacterial and fungal infections, including those caused by Candida auris. Of course, overwhelmed ICUs, suboptimal infection prevention and control, temporary staffing, and of course, people receiving multiple interventions, with indwelling devices, broad spectrum antibiotics, corticosteroids, etc. And sorry, this map just shows you places where COVID-associated Candida auris infections were reported. In South Africa, we really do think that the COVID pandemic accelerated the replacement of Candida auris or of other pathogens by Candida auris. So this is an epidemic curve of cases of candidemia. And I'm showing you 2019 right now. So the green bars indicate the number of cases of Candida auris invasive infection, and the purple line indicates the moving average. So now what I've done is superimposed COVID infections and admissions in South Africa, looking at the 4 major COVID waves that we experienced in South Africa caused by the ancestral virus, the Beta, Delta, and Omicron variants. In yellow are the hospital admissions, and blue are the laboratory confirmed cases. So what you can appreciate is that after each of the COVID waves, there seemed to be an increase in candidemia cases reported nationally. So these are, of course, just ecological data. When one looks specifically at Candida auris and Candida parapsilosis, Candida auris is shown in, I'm just thinking, I think it's Candida auris is in black. I think you can see that more clearly for both auris and parapsilosis in terms of the moving average. These, of course, are both pathogens which are transmitted in an acute healthcare setting. So you would imagine that there would be more transmission post COVID waves where there's increased bed occupancy in these units, in these hospital units. So when we look at our national data, we see that the proportion of Candida auris infections increases between 2019 through to 2022, with a concomitant decline in the proportion of Candida parapsilosis. There's a numerical increase in both auris and parapsilosis, but a much more rapid increase in auris. And you can see that the peak numbers were really seen in 2021. And I really think that this is a replacement of parapsilosis by auris. They both occupy the same ecological niche in a healthcare setting. They've got the same mode of transmission, but Candida auris has some advantages in terms of its much broader resistance to antifungals and disinfectants. We see a similar picture in the public sector, but a much slower replacement in inverted commas. You can see that by 2022, Candida auris accounted for about 9% of all infections, so much smaller proportion than in the private sector. So now let's turn our attention to people infected with Candida auris, mostly with invasive infection. So in terms of the laboratory identification of Candida auris, so we can obviously culture the isolate from a variety of specimens and then identify Candida auris using biochemical methods. And this identification to species level is usually based on a percentage match to assimilation or fermentation reactions. So if the pattern is incorrect, the biochemical test will actually give the incorrect identification rather than no identification. And this has been a problem with Candida auris all along. The Vitek 2 system now gives a reasonably accurate identification with a software update. The software version 8.01 enabled accurate identification of clade 4, but not really of the other clades. And since then, Vitek 2 has been updated to version 9.01. Microscan Walkaway, which some people use, and the BD Phoenix system still produce incorrect identifications because Candida auris is not on the database. The MALDI systems, the Brooker Biotyper, and the Vitek MS both work if they're used with the correct libraries. And they are, of course, then molecular assays. You can also directly detect Candida auris straight from blood culture. So, for instance, the BioFire film array allows direct detection of Candida auris from blood culture, as does the GenMark ePlex ID panel. I'll talk about the chromogenic agar and PCR assays a bit later when we talk about screening for colonization. So Candida auris has been around in South Africa for at least, more than a decade. We reported our first 4 cases in 2012. But when we looked retrospectively at our laboratory-based surveillance data, we found that the first case actually was from 2009. By 2016, 2017, Candida auris was pretty entrenched. You can see that these are the cases of candidemia caused by Candida auris in 2016, 2017. And, 5-6 years ago, most cases were really concentrated in Gauteng province. In terms of the characteristics of people with Candida auris candidemia, they had fairly typical risk factors, including, having previous hospital admissions, being admitted to ICU, being mechanically ventilated, having central venous catheters in situ, previous systemic antimicrobials and antifungals. But interestingly, the length of stay to first positive blood culture is around 4 weeks for Candida auris. Accrued mortality is around 40 to 60%. This is not attributable mortality because these are very sick people. So then we just go back to Chris Hani Baragwanath Hospital, from which I told you about that outbreak notification. This is an epidemic curve again, showing you the number of cases over time between 2016 through to 2020 by ward. So this is for the entire hospital. The colors indicate the ward, red, neonatal, and the window shows you where NICD was notified of a cluster of cases in September 2019. So prior to this, we had a few sporadic cases in the neonatal unit, being shown in red. So you can see a few cases in 2016, 17, 2018. But then really after that outbreak notification, you can see a real explosion of Candida auris in that neonatal unit. And by this time, 2019 through to 2022, I've already told you, Candida auris already becoming a major pathogen, and accounting for 25% of all candidemia cases nationally. And the concern, of course, being that you see Candida auris in red. Now cases of candidemia caused by Candida auris in every single province. And just thinking about neonatal Candida infections, Candida is, just is the number 6 cause of neonatal bloodstream infections and meningitis after various other gram-negatives and gram-positives. We've seen similar proportions of Candida infections before and after COVID, but much higher numbers. So in the 3 years post-COVID, where we've looked at these data, there were more than 2,000 cases versus over 6 years pre-COVID, there were just over 3,000 cases. So in a shorter period of time, we're seeing many more cases of candidemia in newborn babies relative to bacterial causes. And if we just look at Candida species and, within Candida species, you can see that we're seeing a 7-fold increase in the absolute number of auris cases pre and post-COVID, with a 10-fold increase in the proportion and a concomitant decline in parapsilosis. So let's just go back to Candida auris and, just some of the basic epi. So our initial data from about 10 years ago suggested that there was simultaneous independent emergence of Candida auris in 4 regions of the world. The 4 major clades of Candida auris are separated by hundreds of thousands of single nucleotide polymorphisms, so really are very, very different. Each clade probably has spread through clonal expansion, because within each clade there's only one mating type. The A mating type for clades 1 and 4, and the alpha mating type for clades 2 and 3. This study just looks at a more diverse set of isolates from 19 countries, and using a molecular clock analysis, they found that the most common recent ancestor was within the last 360 years. Clade 2 is the oldest of the clades, and clade 4, the South American clade, emerged most recently. And as I mentioned at the beginning of the talk, the drug-resistant strains probably emerged within the last 40 years, when azoles became available in agriculture and human medicine. When we look at the South African picture, this is a whole genome sequence analysis of 115 isolates between 2009 and 2018, including clinical isolates, environmental isolates, so invasive and colonizing isolates from surveillance. Most of these are clade 3 isolates shown in the red circles. Some are clade 1, some are clade 4. Almost all are fluconazole resistant, shown by the pink outer bar, and a few are echinocandin resistant, shown in the yellow outer bar. When we look just at Chris Hani Baragwanath Academic Hospital, we see that there's clade mixing. The neonatal outbreak is predominantly clade 3, and we see clade 4 dominating in both the pediatric and adult burns units. Clade 3 and 4 actually have opposite mating types, and there, of course, is the theoretical risk of sexual recombination if a single person is infected with 2 strains at the same time. In terms of antifungal susceptibility testing for Candida auris, just to start by saying that there are no formal breakpoints endorsed by the major committees or institutions, CLSI or UCAST. We've tended to use CDC tentative breakpoints, which are based on MIC distributions data from animal models, as well as molecular identification of resistant mechanisms. So if we just look at our South African data, looking at both invasive and non-invasive strains between 2018 and 2021, you can see that for fluconazole, the vast majority of isolates have very high MICs, well above the CDC tentative breakpoint of greater than or equal to 32. So resistance is generally mediated by mutations in the ERG11 gene or by increased ERG11 copy numbers. Amphotericin B, the vast majority of isolates have low MICs, now below the CDC tentative breakpoint of greater than or equal to 2, both for invasive and non-invasive isolates. And the few isolates we see that do seem to have higher MICs come from the private sector, and particularly among non-invasive isolates. Similarly with echinocandins, low MICs, both for invasive and non-invasive strains and below the CDC tentative breakpoints. And again, we're seeing a few isolates which have higher MICs, again, in non-invasive, mostly from the private sector. Resistance to echinocandins mediated by mutations in FKS1. There are some specific amino acid substitutions which occur with Candida auris. So we have found some cases where Candida auris is resistant to all the classes of systemic antifungal agents, of azoles, echinocandins, polyenes, as well as flucytosine. And we found these pan-resistant auris isolates mostly from Gauteng province, again in the private sector. Interestingly enough, in the non-ICU setting, and mostly from urine specimens. We've not checked the clades yet, but they probably are all clade 3. In terms of hidden reservoirs of resistance, just to state that resistance can develop, particularly high-level echinocandin resistance can emerge with treatment, so on treatment. So in this particular study, a patient treated who developed Candida auris candidemia with a urinary tract source and with prolonged echinocandin treatment developed Candida auris infection, which was resistant to echinocandins. And I think, similar findings elsewhere, not just restricted to this particular case report. Of concern in the US, transmission documented of pan-resistant strains. The CDC reported a couple of years ago 2 clusters, one in the District of Columbia and another in Texas. There were no epidemiological links between these 2 clusters of echinocandin and pan-resistant strains. None of these patients had prior echinocandin exposure, so it does suggest transmission of pan-resistant strains within healthcare facilities. What about treating pan-resistance? I mean, our first-line treatment for Candida auris candidemia is either amphotericin B or an echinocandin. There are some new agents. Ibrexafungerp is an oral triterpenoid with echinocandin-like activity. It's currently marketed for vulvovaginal candidiasis. It's got very high tissue penetration into the kidney and bladder. And there's a clinical trial currently recruiting into India and South Africa. These data presented, I think, at ECMID last year, or earlier this year, ECMID in 2023, just showing 4 cases, UTI, patients with UTIs successfully, 3 of whom were treated successfully with oral ibrexafungerp. Of course, this is a useful agent, potentially useful agent, because we know that there's limited penetration of echinocandins, for instance, into the lower urinary tract Managepix is another new antifungal agent with a completely novel mechanism of action. It inhibits GWT1 and it has very potent activity against Candida auris. And in a clinical trial which was conducted in South Africa, a phase 2 study, which enrolled a small number of patients, only 9 patients, you can see that 8 of the 9 patients were successfully treated with Managepix. What about combination treatment? So as we see more treatment failure, which is obviously not strictly defined, the opportunity or the possibility of using combination antifungal agents, should be considered. Currently in the South African auris guidelines, we only recommend using combination antifungal treatment in certain circumstances. For instance, for patients with septic shock or patients who've got persistent or recurrent infections. But increasingly, I think people are thinking that if we use combination antifungals up front, could we then preserve those antifungals and minimize the emergence of resistance? And in fact, these data from Exeter show some checkerboard assays, which essentially show that flucytosine, in combination with inidulafungin or Managepix in combination with inidulafungin had very good potency. Just to remind you, there are relatively few systemic antifungal classes we've got access to. We've got the polyenes, the azoles, the candins, etc. And I've already mentioned some of the new antifungals in the pipeline, Managepix, the triterpenoids such as ibrexafungerp, and then of course some modified agents. Some of you might have read about rezafungin recently, which is an echinocandin which can be given once weekly instead of daily, and that's potentially very useful for step-down treatment, but also obviously for first-line treatment. For Candida auris, many of these new agents are completely out of reach. We obviously can't use the azoles, and so we really are stuck with amphotericin B in the public sector, mostly amphotericin B deoxycholate and echinocandins, as well as flucytosine, which is a possibility to be used in combination with the other agents. So I'm going to move now on to colonization. Colonization is a risk factor for subsequent invasive disease. Around 10% of people who are colonized will then develop invasive infection. It's also a reservoir for onwards transmission, as you all know, either directly from person to person or when a colonized person sheds Candida auris into the environment. So could we use a search and destroy strategy, as has been done for MRSA? And has also been considered for carbapenem-resistant Enterobacterales. Though, of course, with CRE, there are many more limitations, both in terms of the screening method and decolonization. So could search and destroy be applied to auris, and in which circumstances? So let's think about the colonization detection methods. So Candida auris is usually colonizes the skin. I'll show you some data shortly. We tend to use amines, agar gel transport swabs, and we use either a culture-based technique to detect colonization or molecular assays. The culture-based techniques are obviously slower. One option is to use sabrode broth containing salt and dulcitol. Candida auris is actually halo tolerant and also temperature tolerant, so high temperature, thermotolerant. So if you use the selective medium, it allows Candida auris to grow where other Candida species can't. Chromogenic agar also works really well. Pictured here is Chroma agar Candida plus. And you can see that the Candida auris colonies are fairly distinctive. These light blue colonies with a blue halo, which is reasonably distinct from other Candida species. There are some false positives, but it's a reasonably good screen. And then, of course, there are molecular assays, PCR assays included amongst those. So how do you search for Candida auris? These are data from an in-depth investigation at a US skilled nursing facility, which was endemic for Candida auris. They conducted 3-point prevalence surveys over a 4-month period from 10 body sites, really to determine the spatial extent of colonization among the long-term care facility residents. And they found that the anterior nares were the most densely colonized site. If you added in the axilla and groin, which are commonly selected sites for screening, this takes the yield up to about 80%. But in order to get to 100% sensitivity, you need to swab many, many more sites. At the same skilled nursing facility, they conducted repeated point prevalence surveys and essentially demonstrated that the prevalence of Candida auris colonization increased over a 10-month period. This was likely due to unrecognized colonization on admission and ongoing transmission within the facility. You can see that the prevalence plateaued at around 70%, probably reflecting some sort of achievement of a steady state between admission and discharge of residents colonized with Candida auris. That's an inpatient setting. These are data from New York City between 2017 and 2019. They looked at colonization post-discharge, and they found that the time taken for patients to become serially negative is around 9 months. For people who become serially negative, the median time to the first negative Candida auris swab is around 5 months. So it seems that once you're discharged from a facility where there's ongoing transmission, you can convert to being negative or colonization negative, but it does take several months. What about destroying Candida auris? I mean, there are no specific protocols for decolonization. And what about recolonization if you're in the same healthcare environment? One option is chlorhexidine, which has been explored in the context of these long-term care facilities where they found a significant reduction in the odds of Candida auris colonization at sites where, body sites, where the chlorhexidine concentration was above a particular threshold. That threshold was 625 micrograms per milliliter, which is a bit arbitrary, but that was the concentration required to get rid of Candida auris. Unfortunately, that was only achieved at 7.3% of the skin sites. So, quite difficult to actually decolonize. There are other options for decolonization, but they've not really been well established. So if we look at the data again at that same neonatal unit, Chris Hani Baragwanath Hospital, Candida auris is now really endemic in that unit. Red here in this epidemic curve shows you invasive infections. The gray-blue color is colonization. I think it's been very difficult to get on top of this despite enhanced infection prevention and control, antifungal stewardship, and environmental cleaning. Now, in 2021, this unit was moving to a new facility. So we took the opportunity at that point to perform a unit-wide point prevalence survey just before the unit moved. The intention really was to identify people who were colonized, babies who were colonized, and to rapidly cohort them. So over the course of the morning, the team, including the on-site team, swabbed every single baby, so 195 babies in that unit on that particular day, and found that the overall prevalence of Candida auris colonization was 33%. 15% of the babies were PCR positive, and those results were available to the unit within 48 hours. The remaining babies were culture positive, and unfortunately, that information was only available about 10 to 14 days later, because culture-based methods are much slower. Nevertheless, those data we used to cohort the babies, the unit then moved to its new facility. And what we did show is that post point prevalence survey and the move to the new facility, there was a reduction in the infection and colonization incidence. Now, these data just go until June 2022. We haven't looked at the force of infection since then. And if there's anyone from Chris Hani Baragwanath Hospital, I'd be interested to hear if anything's changed in 2023. Let's move on to contamination of the environment. Just briefly, of course, I've mentioned that both infected and colonized people can shed skin scales into the environment and contaminate both the environment as well as shared equipment. There was a large outbreak, well, a relatively large outbreak compared to what we see. Between 2015 and 2017 in the UK, I think it was a neurosurgical ICU, 70 patients with auris colonizational infection, also caused by clay 3. The risk of auris infection was associated with the use of these reusable skin surface axillary temperature probes, which had been used in 86% of cases. Candida auris was also isolated from these probes, and these were genetically identical. And the outbreak ended when these temperature probes were removed. It turned out that they'd used quaternary ammonium compounds to disinfect those probes, and you can't really use Candida auris really is resistant to quaternary ammonium compounds. Recent reports, I saw this just maybe a week ago, evidence of airborne dispersal. So this is a Hong Kong cardiothoracic unit. 18 patients had asymptomatic auris skin colonization. They found auris, as you would expect in samples from frequently touched ward items, but then they also found auris in ceiling supply air grills, from corridor air grills, and also from passive air samples, including in a cubicle where there were no confirmed cases for days, suggesting that there was possible air dispersal of Candida auris. And whole genome sequencing confirmed clinality of the air environment and the clinical isolates. I think that's really very concerning. We know that I've just mentioned Candida auris is resistant to quaternary ammonium compounds. It remains viable on surfaces for up to a month. It can be cultured from bedding for up to 7 days. And so patients can acquire infection from the environment within 4 hours of being admitted to a ward where there's contamination by Candida auris. Cleaning really depends on bleach, 1,000 parts per million at minimum. And of course, with terminal disinfection, you can take it up to 10,000 parts per million. Adjunctive methods include UVC disinfection. But of course, there are lots of issues with non-touch disinfection because you really do need contact with the surface. We can talk about the UVC if anyone has any questions. I'm just going to end by talking about surveillance. So of course, this is a satellite photo of Africa at night. It's much less illuminated than Europe to the north. There are a few bright spots, and that's my analogy for Candida auris. When one searches for Candida auris and Africa, you see really very few places reporting Candida auris, either infection or colonization outside of South Africa. There have been case reports from Algeria, Nigeria, Egypt, Sudan, and Kenya. So to this end, we have, to try to really understand the burden of fungal diseases more broadly in Southern Africa, we've set up a fungal disease surveillance project in Southern Africa to create some new cross-border partnerships to understand laboratory capacity for mycology, to design tailored and novel surveillance approaches, and also train people in fungal identification and susceptibility testing, ultimately with the aim of improving and developing, I suppose, fungal genomic sequencing and analysis capacity. This is, the laboratory surveillance pyramid. We use laboratory surveillance very successfully in South Africa, and that's really because, if one looks at people with Candida sepsis in a particular hospital, one needs blood cultures to be collected because, a smaller proportion of all people with Candida sepsis in hospital will actually have a blood culture. A smaller fraction of those will have appropriate lab processing, and the fungus will be detected and identified. Cases need to be collated, submitted, and reported. And of course, you get attrition at each step through the surveillance pyramid. And the attrition is most remarkable in most African countries at the blood culture collection stage, where blood cultures really aren't available. How do you bridge this diagnostic chasm and still understand the burden of Candida auris, I suppose, infection or colonization in Africa? So we've approached this in two different ways. We've conducted point prevalence surveys in acute care hospitals, and we've also been looking at testing wastewater samples, very much as has been done for SARS-CoV-2. Just to give you some updates on our point prevalence surveys, our ultimate aim is to collaborate with 9 countries in the Southern African region. To date, we've completed point prevalence surveys in Angola, Zambia, and Botswana. We've got surveys still pending in Zimbabwe and Mozambique, and we're still planning surveys in Namibia, Malawi, Lesotho, and Eswatini. But you can see that, the first round of point prevalence surveys have already borne fruit. We've managed to detect Candida auris in Botswana as well as in Zambia. And these are the first reports of Candida auris from both these countries. Wastewater surveillance is another approach. These are data from, these are pictures and data from Nevada in the USA. They had their first clinical case detected in 2021. By June 2022, so about a year later, they had 300 cases with 22 facilities affected, and most were clade 3, so the African clade. they looked at wastewater influent samples from 2 treatment plants, and they processed the wastewater using a selective culture medium. They found 2 Candida auris isolates from wastewater, which happened to be genetically related to the clinical isolates in Nevada. So the question is this method sensitive enough to detect Candida auris in wastewater before clinical cases emerge? In the Las Vegas metropolitan area, they've taken it to the next step or the next level. Over a 10-week period, they collected samples from multiple treatment plants in different sewer sheds. They processed the pellets using molecular methods. And the map on the right-hand side shows you sewer sheds. The color is the average influent wastewater concentration of Candida auris. The black diamonds are the 22 facilities reporting cases of Candida auris. And they essentially found a reasonably good correlation with the proportion of facilities with auris cases. And, you know, this really has potential. It could allow you to zoom in strategically to areas where Candida auris is a problem, and could also be used as a method to determine whether interventions are successful. There are obviously issues because we don't really know what the shedding rate is from cases. We don't really know if Candida auris persists in plumbing or sewer systems and biofilms, for example. So it's not perfect. So in South Africa, we've leveraged the COVID wastewater surveillance system. We've been processing wastewater samples over the last 18 months or so. And we've actually found 10 isolates, 10 Candida auris isolates directly from wastewater. 10 of these isolates have been cultured, and of concern, 4 of these have been pan-resistant, resistant to echinocandins, flucytosine, azoles, and amphotericin B. This may imply either that, you know, these isolates were shed from patients who were infected with a pan-resistant strain, or pan-resistance may have been acquired in the environment. I'm going to end there just by summarizing that Candida auris is now a major pathogen, accounting for 1 in 4 invasive Candida infections in South Africa. This has really been amplified by the COVID pandemic. It's now a major invasive pathogen in the inpatient neonatal population. It's emerging in other African countries. Through our surveillance, we've just been able to detect it in Zambia and Botswana. Pan-resistant clinical and environmental strains have been found in South Africa, and there really is insufficient action on the continent to control the spread of this infection. Acknowledgements really to my team at the NICD. This is the team at CHARM, and also our funders. CDC was instrumental in providing funding for the fungal surveillance project. And then, of course, acknowledgements, as always, to GERMS. I couldn't really do much of this work without the GERMS team. And then just to say that, if one looks for Candida, you can find Candida in the most unexpected places. So we have found Candida in Mozambique, but just not in an expected place. Thanks.
Thanks very much. That reminds me of I think it was Chicago restaurant that was Sam and Ella's Chicken, which I thought was pretty great. Fantastic overview, Nelesh. Wonderful, wonderful work, and really exciting to see some of the new data and the new trends. Exciting in one way, worrying in another. A whole bunch of questions have come in, for some on the chat, some to my phone. I guess the first one maybe is are there other known sort of evolutionary advantages which might explain Candida auris sort of overtaking so much of the other species of Candida? I mean, obviously there's resistance, which you mentioned, which definitely obviously helps hugely in a hospital environment. But I mean, obviously the species is, as you said, several hundred years old, and does seem to be overtaking other strains of Candida, even in places which maybe don't have a lot of other azole use. I mean, is it very persistent in the environment more than other Candida species, or does it like biofilms more or something, or do we have any handle on any other advantages it may have, which to explain its sort of evolutionary success?
It certainly does grow in biofilms, Jeremy. That's certainly one of its capabilities. I think that its resistance to antifungals, its resistance to disinfectants, and particularly, I think it's this ability to be shed into the environment and to contaminate surfaces for prolonged periods is fairly unique. We haven't really seen that quite as clearly with other Candida species. I mean, Candida parapsilosis probably is a close second, but it's not. It's a weedy pathogen compared to Candida auris.
And then maybe related to that, in some ways at least, is Graham's question about the 4 different clades arriving in different parts of the world and sort of emerging more or less simultaneously. Do we have any better understanding of this now?
As it turns out, there are now 6 clades. I think we're going to understand more and more about Candida auris. There still are the 4 major clades, but just about a month or so ago, the 6th clade was discovered in the Indo-Malayan area or region of the world. So really in Singapore and Bangladesh. But the 4 major clades seem to arise independently because they really are completely distinct on different continents. We now are seeing phylogeographic mixing. So, in South Africa, as I said, we now see clade 1, 3, and 4, with 3 dominating. But what you really see, and typified by what happens at Chris Hani Baragwanath Hospital, you get a particular clade, establishing itself in a unit. For instance, clade 4 established itself in the burns unit, and it then becomes dominant. It can't be replaced by any other clade. Clade 3 doesn't come in and replace it because it's occupying that ecological niche. And I think that's what you see in healthcare settings. The most successful environmental pathogen, which also is invasive, just takes over. And I think it's in some ways even more successful than Acinetobacter, which is really worrying in some ways.
And the sort of more or less simultaneous discovery, just a sort of coalescence of factors, more healthcare associated environments and more azole exposure and things like that, or is there there's a simultaneousness, which is the weird thing, right? Of these.
I don't think we understand why that happened. People have looked back into the culture collections. There was one isolate, I think, that was seen in South Korea in 1996. But when you look at clinical cases, they all sort of seem to have emerged at around the same time. And it's obviously related to use of azoles. And we can see that the drug-resistant clones, using clones quite loosely, emerged in the 1980s when azoles were broadly used. But I don't think we know why it just popped up all everything popped up all simultaneously.
Okay. A question from Evan about do we have any data of flucytosine susceptibility in South Africa or internationally?
Yes, we do. So we've tested all those isolates I showed earlier against flucytosine. We have almost universal, well, there are no breakpoints, but very, very low MICs to almost all strains. I mean, 0.5% of the isolates were had MICs greater than 16. And that's really useful and interesting. We heard at the Trends in Medical Mycology conference last week that in New York City, or New York State actually more broadly, where they've been using combination echinocandins and flucytosine quite widely, that they've started seeing flucytosine resistance, which is concerning. So you've got to be, if you do use the combination, you've got to be careful about how it's used. If you use the drugs sequentially, there's you have more chance, I think, of resistance. For instance, if you just add a single drug, flucytosine, potentially to a failing regimen, that may not be the best idea.
And then Colleen Bamford's asking about any updated information on the geographical distribution of Candida auris in South Africa. So initially, I know you said it was sort of epicenter was Gauteng. I presume now it's pretty much everywhere, or is that not the case?
Yes, it's in every single province, still dominated by Gauteng, but I think every single province, we've got the data, and I've got the proportions. We just probably need to map it out so it's a bit clearer, and we see it both in the private sector and in the public sector. When we look at our data, 2/3 of all invasive Candida infections are diagnosed in the private sector and 1/3 in the public sector. So it's really all that epidemiology driven by the private sector.
Another question from Evan about are antifungals much used in veterinary health? I mean, is that a potential mechanism of resistance, or do we think it's really just human hospitals?
I suspect it's human use, but of course, there might well be lots of azole fungicides are used in agriculture, and so there's absolutely no doubt that we've got. I mean, there's pretty good evidence that azole-resistant aspergillus emerged because of the use of azole-like agents in the environment. What's really concerning is, I just mentioned that brand new class of antifungal agent typified by manager picks, and already there's an agricultural fungicide which belongs to the same class, and that is now being used. And so, our ability to retain these new classes of antifungals, which are so sort of few and far between, is being jeopardized by this use across sectors.
And then I know it's full, but I'm going to just ask the last two questions very briefly. Nasisa asked about a bit more about the postulated mechanisms for this association between the increase in Candida auris with the COVID outbreaks. So obviously the association is very nicely depicted. It's lovely data, but she was asking what you think drove that specifically.
Nasisa, I mean, it's ecological data, so it means that I can't say it's cause and effect. We're just seeing the association in time and space, and I would just speculate that it's because particularly with the Delta wave, I think that we saw that there was increased bed occupancy during that time, even though people were adhering to infection prevention control measures. I think that some of those measures would have broken down. Some, IPC would have, not been ideal, and so there probably was just increased transmission. And I think once Candida auris establishes itself in a unit, I think we've just seen a persistence post-COVID. And I think, that example of what we saw at the neonatal unit in Krasani Barrow is a good example. It's very difficult to get rid of Candida auris once it's established itself.
And just lastly is a question about mortality in Candida auris. So the person who asked the question, I mean, and you did mention obviously this, it's got a high associated mortality in a crude sense, but it's obviously very, very difficult to tease out how much of that's due to the pathogen versus just the scenario of being in an ICU or invasive devices. Has anyone tried to do that? And do we have any sense of whether it's intrinsically more virulent, let's say, than Candida auris or other Candida species?
Well, I mean, of course, the most virulent pathogens are Candida albicans, because that's the one with most adaptations to human infection, which is why it was always the dominant pathogen. I don't think Candida auris has quite the range of virulence factors as Candida albicans. But, in terms of trying to answer the question of attributable mortality, I think we're very well placed to do a case control study to try to understand what the attributable mortality would be in South Africa. So, it's a good idea. We should absolutely do something like that here.
Thank you so much, Nelesh, and thank you for being so generous with your time. I'm sorry, I know we just slightly over 4, but lots of very good questions which came in, and thank you so much. A wonderful overview, and we've all learned a lot, myself included. Much appreciated. Thank you.
Thanks, Jeremy. Great to be here.